The gain of function research is highly controversial and should be banned and was banned, and it's high risk. You know, again, a dark art in essence.
Well, it absolutely is. Again, in my Bayesian analysis, I talk about the fact that it's absolutely published. For 30 years, there has been on average 0.9, so that rounds to one laboratory acquired infection per year in Asia. So these are SARS-CoV-1, other viruses, one a year. So you're just playing Russian roulette with the 7 billion people on the planet.
There is no good explanation. I'm reading this. This was written by Nicholas Wade, who I interviewed about a week ago. There is no explanation of why a natural epidemic should break out in Wuhan and nowhere else. There is no good explanation of how the virus acquired, and I'm hoping you can explain this, its furin cleavage site, which no other SARS-related beta coronavirus possesses. What does that mean?
Yeah. So it's one of my 5 facts that I talk about that are not in dispute. So there are 5 facts about this event that every scientist agrees to, but they all lead to the conclusion it came from the laboratory. So one of them is this unusual furin site. Okay, so the spike protein is how the virus gets into cells. It's basically like a key, and the lock that the cell has is called ACE2. So it's a protein on the surface of your lung cells, and the spike protein binds to it. And then a second step has to happen where the other part of the spike protein makes a hole in your membrane and injects the RNA. And then it takes over the cell, and then it makes a million of its own copies. So in order for this, the spike protein is one big protein. So in order to do that physical process of injecting the RNA, it has to actually physically move, and the binding part, you got the binding part to the ACE2, and you got the other part here. This part has to physically move like 100 nanometers, which is a huge distance on a molecular level, to let the RNA go into the cell. So the spike protein has to be clipped. So most beta coronaviruses clip it at a place called S2-1. There's only a couple enzymes that will clip it there, which means there are only a couple kinds of cells that it can go into. So we've known since 1992, 11 different laboratories have done it. Dr. Barrick and Dr. Shi at the Wuhan Institute. Since 1992, 11 labs have put furin sites in coronaviruses. And 11 out of 11 times, it does one thing. It makes it either more infective, more transmissible, or more lethal, or all of the above. And the reason is it gives a new way of activating that spike protein with an enzyme called furin, which is our enzyme. So basically, the virus is very economical. It doesn't bother having an enzyme to activate itself. It knows with this new furin site that it'll be able to get into a cell.
We've never found furin cleavage sites in natural occurring proteins, or what do you want to call it?
So look at so the virus kingdom is very big. So influenza uses them all the time. Certain other viruses use them all the time. Beta coronaviruses never seem to use them. So we have over 2,000 sequences of beta coronaviruses from nature, and the number of furin sites is 0. Well, it's 1 is SARS-CoV-2. So there's no natural furin site in any beta coronavirus. That's just, I mean, look at 0 out of 2,000. You ask a statistician what the incidence in the population is, and it's under 0.1%.
It tells you what about COVID-19, that furin cleavage site?
So it has it.
And it tells you what about the fact that it has it?
Okay, so if there's, it doesn't.
It tells you it was lab created.
Well, yes, so basically, yes, it's never been found in nature in that class of viruses. And in 11 laboratories since 1992, it's purposely been put including in coronaviruses, to make them more infective. So that's one layer of the furin site, which makes it special. I call it the immaculate cleavage site, because it literally has to be if it came from nature. The next level is the genetic level. So the furin site is 4 amino acids in a row. Now, I worked with Govind Khurana at MIT, who got the Nobel Prize for the genetic code. So there's 3 bases in the code for every amino acid. So it's redundant. So your genetic blueprint that makes proteins has 3 nucleotides, 1 amino acid, 3, 1, 3, 1, 3, 1. So there's 6 different 3-letter words for one of the amino acids in the furin site called arginine. There's 6 different words for the same amino acid. It's redundant. Humans like a particular 3-letter word, CGG. Bat coronaviruses hate CGG. Out of the 64, so if there's 4 nucleotides and 3 letters in a codon, that is 64 different possible codons. So the least popular codon for coronaviruses is CGG. They hate it. They absolutely hate it. And yet here it is. Here are 2 of them together. Now, I've looked at 580,000 codons in beta coronaviruses with the help of a computer, and there is not a single example of the 2 together, CGG, CGG, next to each other.
Back to Nicholas Wade's last line, where he says the natural emergence theory battles a bristling array of implausibilities.
Yeah. So I've been working with Nicholas for about 6 months weekly, with some of the science behind some of his writing. So yes. So that's absolutely weird. Now, why would the flip side of that is there a laboratory reason why you'd put CGG in? Because if you think about it, if you've got 6 possibilities for the first arginine, 6 for the second, that's 36 combinations, right?
You mean why as a scientist would you put it in?
Yes, exactly. Why as a scientist would you put it in?
Okay, why?
So we know that nature is never going to put it in. In 580,000 codons, it's never appeared there before. So nature hates it. Why in the heck would a scientist do it if there's 36 possibilities?
Answer?
It's very simple. There is an old technique where if you inserted something genetically and you wanted to follow it in your experiments, you would put in what's called a restriction site. So you'd put in a set of amino acids that gets clipped by an enzyme you can buy off the shelf, and then when you run a gel, if the protein if it's still in there and intact, the protein runs very high on a gel, and then if it's been cut accidentally or it's been lost or something, you don't see it. So it's a very common laboratory technique. The Wuhan Institute's been doing it for years, where when you do an insertion, you purposely put in one of the restriction sites so you can follow it in the laboratory. That's the only one of the 36 that has a restriction site like that. So a lot of motivation for putting that in there.
So a lot of the allegations have been that the US National Institute of Health provided funding for gain of function experiments in Wuhan. And then you had this very telling Senate Intelligence Committee investigation last week where Republican Senator, Rand Paul then, gets in this, what looked almost like a court examination with Dr. Fauci, who was the head of the National Institute of Health. Will you, in front of this group, categorically say that COVID-19 could not have occurred through serial passage in a laboratory? Fauci, I do not have accounting of what the Chinese may have done. I'm fully in favor of further investigation of what went on in China. However, I'll repeat again, the NIH and the NIAID categorically had not funded gain of function research to be conducted in the Wuhan Institute of Virology. Does that ring hollow?
I think it does. I mean, and it may be, in a really lawyerly court-like way, there's a technicality. So what we have is we have the grants that EcoHealth was funded, and we have the subcontracts to Wuhan Institute of Virology inside those grants. So we know exactly what was expected of the WIV inside the grant that EcoHealth got from NIAID, which is Dr. Fauci's work. It's clearly gain of function research. Now, the money didn't go from NIH to Wuhan. It went from NIH to EcoHealth to Wuhan. If that really is what he's relying on, it's disappointing. But I don't have another explanation. Now, he actually also said during that interview that there was no gain of function research being done. And I think on the face of it, on the face of the documents that are available to the public, you don't have to even do a FOIA request. You can just go online in the NIH. You can see they're describing gain of function research in the 5-year grant that was funded, that funded the Wuhan Institute.
They
in
don't call it
2000.
gain of function.
They don't call it gain of function, but they describe what they describe is gain of function. So the gain of function is a very broad definition of just increasing the transmissibility, lethality, or infectivity of a virus in the human population, and using humanized mice and in improving in purposely causing testing to test for spillover potential, is exactly that. I mean, it's, again, I will try to pretend to be interested in what they were doing. So the idea was how close to SARS-CoV-1. So we know CoV-1 spilled over. And then we know that there are these viruses that are 60% similar and 70% and 80% and 89%. Literally what they were saying is, what percent does it have to be like SARS-CoV-1 so we know it spills over? And then we can use that number to go out in nature and say, well, here's a virus that's 85, so it's not likely. Here's one that's 92, so it's likely. Giving them the benefit of the doubt, that's probably what they were trying to do. But we know what the outcome is.
Stephen, just a couple of quick questions as well before I let you go. The data that was online, some of it from the Wuhan lab, some of it that you required a password, I understand, all disappears in September. It's taken down in September. So October, November, some 3 months before the official outbreak, although a lot of people think that they already had cases well into November. What do you make of that, and how important is that data?
Critically important. I mean, 3 a.m. September 19th, a server that was available for the world for most of it, and it also did have a password protected part, but Steve Quay could have gotten on it the day before, but it's taken offline. Now they slipped up and they said, well, it's taken offline because of hackers around the COVID infection. And then they realized, oh my gosh, this was in September. So I'm not sure they're still saying that.
That's a pretty big slip up.
Well, it's out there. I mean, that's how it is with cameras. Anyway, but and it's not available to this day. So I asked Peter Daszak in a public forum here maybe 2 months ago, where if he would provide access to those files, and he literally said it's on the record there that he knows what's in them. He's looked at them, and there's nothing there of interest. And that's the basis for not providing them.
Well, then show them to the world, because right now that lab is under. Look, I mean, I think you tell me if this is an overstatement, but it seems to me that there is a consensus amongst the scientific community now that at the very least there are huge, gaping gaps in the Chinese official story on that lab. So if you want your lab, if you really want to support your suggestion to the world that it didn't originate in the lab, then make all the data available. That's the best thing that they could do right now to the WHO or whoever.
Agreed. Whether they would do that or not is a political question.
Right. Okay. So but that data does still exist, we think.
Oh, I hope so. I hope so.
Are we ever going to get to the bottom of this?
Well, see, that's an interesting question, because with my analysis, all of my evidence is circumstantial, but I remind people that, there are many sort of murder trials in which all of the evidence is circumstantial, and yet we quite routinely can determine the guilt of someone with a proper process and adjudication and getting to beyond a reasonable doubt. So despite it being circumstantial, I believe it's strong enough at this point in time to conclude, again, beyond a reasonable doubt, that it came from the laboratory. The key thing to remember with these coronavirus pandemics is there's 3 components. There's the intermediate, there's the host, the animal host, the humans, and the virus. This is the only one in which it's a singularity. That is, there seems to be only one animal, because we can't find any in nature. So there's one animal. All the human cases go back through one patient at the PLA hospital in Wuhan. So when President Trump got it, you can follow the mutations to his virus back to the PLA hospital. So there's a singularity there. And the sequences inside the virus. The patient. There's absolutely no patients before December who had any hint of this infection. So it is a true singularity.
Which I understand is quite interesting.
One animal. One patient, one virus, one animal. And
because normally it takes a virus time to become more and more transmissible. there should be a trail of patients.